Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2013. If you are new to my channel, please do like, share and subscribe. Coming to June 2013. You have created a fusion between this is part B questions. Between the TRP operon which encodes the enzymes for tryptophan synthesis under the regulatory control. So the regulatory portion is from the lag operator. Under which of the following conditions will tryptophan synthase be induced in the strain that carries the chimeric operator fused operons? So the regulation is from the lag operator side. The enzymes are not beta-galactosidase permease, but they are tryptophan biosynthesis genes. So when will this tryptophan synthesis will be induced is only in the presence of lactose when glucose is absent. So the answer here is option 4. Then next one. Origin of replication usually contain. So they usually contain AT rich sequences. Why? Because if GC rich sequences are present, they will not be broken down easily, because it is easier to break 2 hydrogen bonds than 3 hydrogen bonds. Then sigma subunit of E. coli RNA polymerase does not initiate transcription and fall off during elongation. That is true. Increase affinity of the core enzyme to the promoter. That is also true. Binds to DNA independent of the core enzyme. No, it is dependent on the core enzyme. So that is not true. Option 3. Ensure specificity of transcription by interacting with the core enzyme. That is also correct. The next one. The cap binding protein EIF4E, which is involved in the global regulation of translation, is highly regulated in eukaryotic cells. In an experiment, a researcher transfected mammalian cells with EIF4E gene for its overexpression. So he overexpressed the gene which is required for regulation of translation. So what will happen is that you will get cancerous cells or neoplastic transformation. Answer is option 2. Coming to part C. Phosphorylation of serines as well as methylation and acetylation of lysines in histone tail affect the stability of chromatin structure above the nucleosome level and have important consequences for gene expression. So phosphorylation of serines and methylation and acetylation of lysines in the histone, it will result in loosening of the histone and the loosening of histone and DNA binding to histones. It will result in loosening of DNA binding to histones. So what will happen is that it will result in gene expression. So the resulting changes in charge are expected to affect the ability of the tails to interact with the DNA because. So fourth option, if you see, is methylation and acetylation of lysine increases DNA histone interaction. They decrease DNA histone interaction. Phosphorylation of serine increases DNA histone interaction. Phosphorylation of serine also decreases DNA histone interaction because phosphorylation of serine results in negative charge and it will break the DNA histone interaction. DNA histone interaction is independent of net charge. It is dependent on the net charge and DNA is negatively charged. That is the correct answer. So the resulting changes in the charge are expected to affect the ability of the tails to interact with the DNA because DNA is negatively charged. The next one. You are working with an in vitro eukaryotic transcription system which produced both capped and uncapped mRNAs. You incubated these mRNAs with mammalian cell nuclear extract and then quantified the different products as shown below. Which of the following graphs correctly represent the expected result? So you have got capped and uncapped on the x-axis, and they have given this white box as pre-mRNA. This is spliced mRNA with diagonals, and fully darkened one is the polyadenylated mRNA. So what is the consequence that you will get over? What is the result that you will get over here? So in the capped condition, pre-mRNA will be less and spliced and polyadenylated mRNA will be more. So if you see your option 1, you can see that pre-mRNA is less, spliced and polyadenylated mRNA is more. Whereas in others, pre-mRNA is more, so that is wrong. Pre-mRNA is less, but polyadenylated mRNA is also less, that is not correct. Pre-mRNA is less, but polyadenylated mRNA is more, but spliced mRNA is less. So that is also not correct. So option 1 is correct. But in case of uncapped, you will see more of pre-mRNA and less of spliced and polyadenylated mRNA. So the option here is option 1. Then next one. During elongation step of protein synthesis, translocation moves the mRNA and the peptidyl tRNA by one codon through the ribosome. Translocation in E. coli involves GTP and EFG. However, in vitro translocation can take place independent of GTP and EFG. Based on these observations, the following hypothesis can be made. So, which hypothesis is more correct? The molecular mechanism of translocation in vitro is completely different from that of in vivo. That is not correct. Translocation activity is independent of GTP hydrolysis. That is also not correct. Translocation activity is completely dependent on GTP and EFG. That is also not correct. Even in the absence of EFG and GTP, translocation is taking place in vitro conditions. Translocation activity is inherent in ribosomes. However, the rate of translocation in vivo is enhanced significantly in the presence of GTP and EFG. That is correct. So, which of the following combination is correct? Only D is correct. Answer is option 1. Next question. DNA methylation plays an important role in the transcription regulation in vertebrates. There is an inverse correlation between the level of DNA methylation in the vicinity of a gene and its transcription rate. So when DNA methylation increases, transcription rate decreases. That is what they meant by that sentence. Whereas there is a direct correlation between histone acetylation and increased transcription. So histone acetylation increases means it increases transcription. Now, beta thalassemia is a common genetic impairment of hemoglobin beta chain synthesis in humans. If these patients can synthesize hemoglobin F instead of hemoglobin beta chain in its place, they would be notably benefited. So what they did was administration of 5-azacitidine to beta thalassemia patients increases hemoglobin F level in erythrocytes and thus benefit these patients. So what does this 5-azacitidine do? Which one of the following statement about 5-azacitidine is not correct? So cells exposed to 5-azacitidine incorporate it into DNA in place of cytidine. Now 5-azacitidine, it is an analog of cytosine, so naturally it will be incorporated in DNA in place of cytidine. 5-azacitidine decreases DNA methylation. Yes, that is why beta thalassemia patients, there is increased level of hemoglobin F level in the erythrocyte and thus benefit the patient in case of patients with beta thalassemia. So if there is decrease in DNA methylation, there is an increase in the transcription rate. So transcription of hemoglobin F level increases in the erythrocytes in patients with beta thalassemia who have been administered with 5-azacitidine. Then 5-azacitidine It promotes histone acetylation. Yes, because that is why there is gene expression. As already mentioned in the case of DNA methylation, because it promotes histone acetylation means transcription rate increases. 5-esa-cytidine does not promote gene expression. That is not correct. So they have asked for the one which is not correct, and your answer is option 4. Coming to December 2013. In bacteria, chromosomal DNA replication starts at one specific locus. Then UV or ABC repair mechanism is involved in repairing DNA damage caused by bulky chemical adducts. Then which of the following is not a post-translational modification in a mammalian system? It is peptidylation. Answer is option 3. Next. E. coli proliferates faster on glucose than it does on lactose because lactose is taken up slowly than glucose. E. coli has got more preference for glucose than for lactose. So when glucose and lactose are present together, it will easily take up glucose, whereas lactose is taken up slowly than glucose. So option 1 is the answer. Then next one. In a cell-free extract containing DNA polymerase 1, Mg2 positive, DATP, DGTP, DCTP, DTTP, H3, the following DNA molecules were added. Single-stranded closed circular DNA molecule containing 824 nucleotides. So before coming to this statements, one thing I have to tell you is that DNA polymerase 1 is template dependent, and it synthesizes in the 5' to 3' direction, and it adds nucleotide to the 3' OH group. Now single-stranded closed circular DNA molecule containing 824 nucleotide, that is like this, as shown in this photograph. There is no 3' OH there. So synthesis will not take place. Single-stranded closed circular DNA molecule having 1578 nucleotide base paired with a linear single-stranded DNA molecule of 824 nucleotides having free 3' OH group, like this. So there is a closed circular DNA molecule which is single-stranded and 1578 nucleotides long. It is base paired to a single-stranded linear DNA molecule of 824 nucleotides having 3' OH group. So DNA polymerase 1 will synthesize. So there will be synthesis. C. Double-stranded linear DNA molecule containing 1578 nucleotides having free 3' OH group at both the ends. So this is it, double-stranded DNA molecule having 3' OH groups at both the ends, but there is no template for that to synthesize. So that also will not be synthesized. C also will not be synthesized. Double-stranded closed circular DNA molecule having 824 nucleotide, that is like this, there is no 3' OH group. So it will also not be synthesized. With the rate of DNA synthesis was measured by incorporation of H3 thymidine in the DNA molecule and expressed as the percentage of the DNA synthesis relative to total DNA input. Which of the following graphs represent the correct result? So out of these, only B is showing the synthesis. Now this portion that I have drawn here. Okay. So A is circular form, B is triangular form, C is shaded circle, and D is shaded triangle. So out of all these 4 options, only B is being synthesized. So if you see option C, only B is synthesized. That is triangles you see over here. So B only synthesis is taking place, whereas in other synthesis is not taking place. So the answer here is option 3. That is this one. Okay. Then next one. The complex responses to different types of DNA damage in prokaryotes and eukaryotes fall into three main categories. Damage bypass, damage reversal, damage removal. Many repair proteins are isolated like DNA methyltransferase, DNA glycosylase, DNA polymerase 4. Which of the following represent the correct combination? So, 1 is C, DNA polymerase 4. 2, A, DNA methyltransferase. And 3, DNA glycosylase, that is damage removal. So the answer here is option 3, 1C, 2A, 3B. Then next one. In recent years, genome-wide transcription study using high-throughput sequence analysis has revealed some novel results that include presence of RNA polymerase in both intra and intergenic region of the genome. Intra is your coding region, intergenic is non-coding. Okay. So presence of RNA polymerase in both intra and intergenic regions of the genome. Existence of non-coding RNAs generated from mRNA coding genes. Existence of sense and antisense transcripts generated from the promoter and untranslated region of many annotated genes. Possible interpretation of the above results are: RNA polymerase can loosely bind to any part of the genome, but its affinity becomes strong only when it reaches the promoter. So that is not correct. Then next one. Binding of RNA polymerase to non-promoter regions of the genome leads to the generation of various non-coding regulatory RNAs. That is correct. Non-coding RNAs are generated from mRNA coding genes due to aberrant transcription initiation and termination. That is also not correct. Sense and antisense transcripts are generated from the promoter and untranslated regions of the protein coding genes by a novel mechanism of bidirectional transcription. That is also correct. So the answer here is option 2, that is B and D. Then next one. Amino acyl tRNA synthetases face two important challenges. They must recognize the correct set of tRNAs for a particular amino acid. They must charge all of these iso-accepting tRNAs with the correct amino acid. Both of these processes are carried out with high fidelity by the following possible mechanism. The discrimination ability resides predominantly at the acceptor stem of the tRNAs. That is correct. The specificity is contributed by the anticodon loop in tRNA. That is also correct. The specificity is embedded in the amino acyl synthetase at the N terminus. That is not correct. The specificity is contributed by the variable loop. That is also not correct. So which of the following is correct? A and B are correct. Answer is option 1. Then next one. During heat shock, mammalian cells shut down global protein synthesis while inducing heat shock proteins. The possible molecular regulation that could explain the phenomenon are. So when there is a heat shock, the mammalian cell, they shut down protein synthesis whereas they induce heat shock protein synthesis. So the possible molecular regulation that could explain the phenomenon are mRNAs of all proteins except those of heat shock proteins undergo degradation during heat shock. mRNAs of all proteins except those of heat shock proteins undergo degradation during heat shock. That is not correct. Cap-dependent translation of most mRNAs is affected during heat shock due to denaturation of cap-binding protein ELF4E. That is correct. Translation initiation of HSP mRNA take place through their internal ribosome entry sites. Already I have discussed that IRS is involved in translation under adverse condition. So C is correct. So the answer here is option 2, that is B and C.